All right, welcome back to General Virology. Today we're talking about Chapter 5, which is primarily all about virus transmission. We will touch just a little bit on some epidemiology as well. And I'm going to have this lecture divided into two videos. In the first, I'm just going to focus on point one here, which is all about how viruses gain entry to the human body. And then in the second half, I'll talk briefly about, well, once the virus is in, how does it move within an infected host? Um, how does a virus exit and then spread to an uninfected individual? That'll kind of lead into talking about some common patterns of infection. And then, as I mentioned at the end, we'll touch briefly on some epidemiology and talk about some basic epidemiological studies. All right, so before we get into portals of entry, let's just talk about virus transmission here for a second. So for a virus to be quote unquote successful, and this is success from the virus's perspective, not from the host, the virus has to be transmitted from individual to individual, and that means successfully initiating infection. And so to do that, there would have to be a certain number of virions capable of entering an uninfected host. Now, theoretically, just a single virion would be enough to establish an infection, but in reality, you have lots of barriers, and we'll talk about some of the different barriers for these various portals of entry. But these could be, mechanical barriers, chemical barriers, immunological barriers, right? There's all these factors that the virus would have to overcome to be able to start that infection. So typically speaking, there's going to be some minimum number of virions that you would probably need to be exposed to in order to actually become infected. And that may vary from person to person or from virus to virus. But essentially, the bigger the dose of virions that you're exposed to, the more likely you are to become infected with any given virus. So we need that exposure. We have these barriers to overcome. The virus is also going to need an accessible host, just meaning it has to come into physical contact with the host, right? Host and virus have to interact. There has to be that accessibility. And the cells in that host have to be susceptible. And this primarily comes down to something we've already talked about, which is are there cells in that host or potential host that express the appropriate cell surface receptors that the virus can bind to in order to gain entry? Because remember, the virus not just has to come in contact with the host, but actually has to gain access to the inside of its cells. And then finally, those cells have to be permissive, meaning they have to contain the appropriate proteins and molecules inside the cell that the virus needs to complete its replication cycle. I'm not sure why this is number one here. I don't know what happened, but anyway, you get the idea. So permissivity is, are the host cells capable of allowing the virus to complete its replication cycle? And this goes back to the fact that viruses are relying on host cell components in order to carry out their replication. All right, so let's talk about some of these various portals of entry. How do viruses typically gain entry into human hosts? And you can see the common portals of entry listed here: the eye, the respiratory tract, gastrointestinal tract, the skin, genital tract, and then we're going to consider congenital infections as well. And so really for the majority of these portals of entry, viruses are typically initially going to interact with epithelial cells. So the epithelium is that layer of cells that covers, the entire outside of your body, but also pretty much all your organs and inner cavities are also lined with a layer of epithelium. So that is usually what viruses are first going to encounter, with some exceptions. There's different types of epithelium. So, for example, there's mucosal epithelium, which is lined in all internal surfaces, including your respiratory tract and your genital and gastrointestinal tracts. And, that, as that name implies, these are epithelial cells that are producing mucus. There are some portals that could bypass epithelium if you have, a wound, so the virus is able to penetrate directly through the skin. If you have a transplant or a blood transfusion, or transplacental infection, which would be occurring through the placenta, and we'll talk about that here in this chapter. It's a little bit more detail. So what I want to do is just go through these common portals of entry, just kind of remind you of the basic anatomy of what's going on there, and then talk about what are some of the challenges that viruses might encounter trying to gain entry through these different portals, and maybe give you some examples. What are some common viruses that tend to infect people through specific portals of entry? So let's start with the respiratory tract. That's a common one. We all think about that, especially now in the time of COVID-19, right? And just to remind you, your respiratory tract is divided into the upper and lower tracts. Upper basically being everything in your head and neck, your nasal cavity and pharynx and so on, and the lower being everything in your chest, your lungs primarily, but also your trachea as well. And so there are a few obstacles virus has to overcome in the respiratory tract. These are, of course, mucosal epithelia lining that. So virus could become trapped in the mucus. You've got cilia sweeping debris, including potentially virus particles, out of your upper respiratory tract. You've got the flow of air and the flow of mucus that could sweep virus out. And then you've also got antibodies and macrophages, which are phagocytic types of white blood cells that are prowling the respiratory tract that could either directly destroy or target viruses for destruction before they really gain a foothold. And so, a lot of common viruses tend to gain access through the respiratory tract. Flu virus, common cold, measles, mumps, rubella, and obviously COVID-19, right? We're all wearing masks and trying to distance in an effort to try to avoid that respiratory transmission. Second port of entry is the gastrointestinal tract, or aka the digestive tract, which I think we're all fairly familiar with that basic anatomy. One thing you might not be aware of, I did want to point out, is in the small intestine you have these structures called Peyer's patches, which are located just below the intestinal epithelial cells. And these are basically lymph nodes that would contain immune cells, including antibody-producing cells, B cells. We'll talk about that in the immunology chapter coming up. But sometimes viruses can gain access through these specialized epithelial cells called the M cells. So these are basically allowing transport of materials through the epithelium in certain instances. So virus could cross the epithelial layer through the M cells, but then they're going to have to evade the immune cells that would be associated with those Peyer's patches. But that is a possibility. Some challenges for the virus in the gastrointestinal tract, of course, is a very acidic environment. And so if a virus is acid labile, that means it's not going to do well in that low pH environment and is probably going to be destroyed. Some viruses are acid resistant, have capsid proteins that don't get denatured in that acidic environment, and might be able to survive the passage through the gastrointestinal tract. So you have the acidity, you've got the flow of food and water, you've got mucus that could trap viruses as well, and we've got antibodies to deal with. So actually your gut is, as we just alluded to with the Peyer's patches, is a key location of immune cells in your body. When we think about gastrointestinal tract transmission from one person to another, one common way this could happen would be infants feeding on breast milk from their mother. So you can, in some cases, have a virus that can actually enter the breast milk and could be passage that way. So, for example, HIV, cytomegalovirus, West Nile virus have all been shown to be able to be passed in breast milk. The other common type of gastrointestinal transmission, person to person, would be the fecal-oral route. So, in places where you don't have proper sanitation, water might be contaminated with feces, or maybe you have somebody preparing food, a restaurant worker that's not practicing proper hygiene. So you could have virus present in feces that could then be ingested by another individual. And, there's a few common viruses that you may have heard of transmitted this way. So a couple of hepatitis viruses, Hep A and hepatitis E. Poliovirus was known to be transmitted this way. Rotavirus and Norwalk virus as well. Next port of entry is the genital tract. I won't go through all that anatomy. I think we're somewhat familiar with that. But primarily here we're talking about STDs, right? Sexually transmitted diseases. So, this is going to be just through direct contact, sexual contact, obviously. Common examples would be HIV, HPV, herpes simplex 2, which causes genital warts, hepatitis B and C, all transmitted commonly through the genital tract. Another big portal of entry, literally a big portal of entry, is the skin. So we have a pretty large surface area, 1.5 to 2 square meters, right, completely covered with skin. And just to remind you, the skin is made of 2 layers. You've got the epidermis and the dermis, epidermis being that tough waterproof barrier, the dermis being the thicker layer that contains hair follicles, sweat glands, etc. And then below that, you've got subcutaneous tissue that has fat and connective tissue. So it's a pretty formidable barrier. And if we just zoom in on the epidermis here, we can see it's actually composed of 5 layers itself. So I'm not worried about going too in depth into this anatomy here. But just keep in mind, you have cells being produced down here in the basal layer. These would be dividing cells. And as these cells are produced, they're sort of being pushed up towards the surface, and as they go, they're basically differentiating into the squamous epithelial cell, which are basically just tough, scaly, essentially dead cells, right, that are constantly being sloughed off. So you do have this tough, keratin-filled, waterproof barrier that is going to be difficult for viruses to gain access to. And as I mentioned, these cells are constantly being lost, so if a virus attaches to one of these cells, there's a good chance it's just going to be discarded pretty quickly anyway. But a few other barriers, you've got perspiration, fluid on your skin that may wash virus particles away. You've got oil called sebum that's very acidic, so that could be a challenge if similar to what we were talking about in the gastrointestinal tract, that could destroy some virus particles. And I was just talking about that outer layer of cells being essentially dead. So you have these non-living cells that viruses are first coming in contact with. So, of course, they can't support viral replication, even if virus was able to gain access to the interior of those outermost epithelial cells. So it's pretty tough, literally, way for the virus to gain entry. But that being said, obviously we get cuts and abrasions on our skin, giving access to some of those lower dividing layers, living cells. So virus could gain access through that route. Insect bites could give penetration into the epidermis to some of the lower levels, or animal bites or needle punctures could completely give access, like directly into the subcutaneous tissue or into the bloodstream even directly, essentially bypassing the skin in that instance. So a few different examples of viruses. Those coming just direct skin to skin contact would be like HPV or herpes simplex 1, which causes cold sores, oral herpes. Um, Penetration into the dermis through cuts or injection would be things like HIV, Hep B, and Hep C. Um, and then you've got some of your like insect-borne diseases, mosquito or tick-borne, like dengue or West Nile or Colorado tick fever is another example there. All right, so we've got the skin, we've got the eye. So obviously the the eye is fairly easy to gain access to, but it is pretty rare to have infections in the eye themselves without some kind of injury, puncture. So I think because our eyes are constantly being lubricated with fluid, that tends to wash away potential viral invaders. So it is possible to have an infection in the cornea. Particular herpes viruses have been known to infect the cornea, which can lead to blindness. Cornea here. A lot of us may have had pink eye conjunctivitis, which is an infection of this thin layer of cells down here called the conjunctiva. That could be caused by different viruses, but commonly adenovirus. And I wanted to mention this. You may have heard talking about COVID-19. Don't touch your face, don't touch your eyes. And there is some preliminary evidence that COVID may be transmitted through the eye potentially. So this was some early study in 2019 out of China. And I won't go into all the details here. You can read the abstract if you want, but basically their key conclusion was it is possible to transmit via the eyes. So yeah, try not to touch your eyes. All right, I mentioned the placenta already. Let's go back and talk about this. So I'm sure you're familiar with the basic idea of the placenta, but it is fetal tissue, right, that is essentially acting as a filter. You can see zoomed in here. That's allowing transfer of gases, oxygen, carbon dioxide, waste products, nutrients between mother and fetus. And so there's actually no mixing of maternal and fetal blood, but we get very close contact between the mother's and the fetus's bloodstream, so you can actually get diffusion from one to the other, and in some cases virus particles may be small enough that they could actually diffuse from mother to child. If that occurs, this would be considered a congenital infection, right? So this covers viruses present at the time of birth, either transmitted transplacentally or from mother to child as the baby is passing through the birth canal. If the transmission occurs in the birth canal, this would be considered an intrapartum infection. And yet another related term to mention is perinatal. So this is going to be infection that occurs either within those weeks right before birth, so typically from 20 to 29 weeks, or even in the first few weeks right after birth. From mother to child, that would be a perinatal infection. And, no matter what we're talking about, intrapartum, perinatal, these infections can be very severe, even with what might otherwise be mild viruses in adults, because, of course, infants' immune systems are not fully developed, and, so it could cause very, very severe issues, including potentially death of the fetus or infant. So while we're on this topic, I wanted to mention this distinction between horizontal and vertical transmission. And so what we were just talking about, transfer from mother to child, would be considered vertical transmission, right? Basically from one generation to the next through congenital or perinatal infection, whereas a horizontal transmission would be from host to host, right? Not associated with mother to child specifically. All right, so by far the most common cause of congenital infections is this virus called human cytomegalovirus. Happens in about 2.5% of live births. This is a virus that, as I just alluded to, does not cause big problems in adults, but can again have some very serious symptoms in infants. Um, a few others worth mentioning: measles, rubella, herpes simplex 1 and 2, variola virus could also be transplacentally uh, passed from mother to child. Let's talk for a second about transplants. So this is not the most common way to get a viral infection, but of course viruses could be passaged in transplanted tissue. And by far the most common transplanted tissue, we don't often tend to think of this way, is blood. So blood transfusions would technically be considered transplants. Um, and there is this whole class of what are called TTIs, transfusion transmitted infections. Um, and so, luckily we've started screening the blood supply for the vast majority of high impact viruses, at least those that we have tests for. Um, but this was not always the case, or in the case of like new emerging viruses, where maybe there wasn't a test initially, this can still be a big problem. So, for example, in the 1980s, when HIV first started to become widespread, they estimated that about 20,000 people contracted HIV just from blood transfusions alone. And so, probably the hardest hit of that population were hemophiliacs in the United States. That were getting regular blood transfusions. Um, so obviously, HIV is a big one on the list of viruses transmitted this way, but also hepatitis B and C, even West Nile virus. So we got blood transfusions, and of course viruses could be transmitted in solid organ transplants as well, but those are much more rare in general compared to blood transfusions. Um, but they can be particularly severe because if you get an organ transplant, you're usually given immunosuppressive drugs, right, to try to help prevent your immune system from rejecting that transplanted organ. So if you do have an infected organ, right, that's being introduced into an immunocompromised individual. So that could lead to very rapid disease progression. And there's a pretty long list of viruses here that have been known to be transmitted in solid organs from time to time. So I'm going to stop there, and I'll pick this up in part 2.